Okay, let's finish part 1 of chapter 3, aerobic metabolism. We're going to finish talking about the Krebs cycle. So I'm going to go ahead and share my screen to where we left off. Okay, so we had pyruvate, right? So after pyruvate, we know that we have to turn it into acetyl-CoA. We already went through that process of looking at the combustion or oxidation of pyruvate and how we make acetyl-CoA. Acetyl-CoA combines with oxaloacetate, and then oxaloacetate forms citrate. Citrate goes through a series of reactions. So we will have citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and back to oxaloacetate. So oxaloacetate begins and ends the cycle. So it's regenerated after those 8 steps. So that's why it's a cycle, it's a closed loop. So let's look at what we produce. For each acetyl-CoA, remember we have 2 of them. These are our products here. We're going to get 2 carbon dioxides. Those are our waste products. We have 1 ATP, 3 NADH, 1 FADH. And so this is kind of basically our overview of what's about to happen. When we look here, I'm just going to explain the red arrows and kind of these 8 steps and kind of what happens. All right, so we talked about acetyl-CoA, and it's going to combine with oxaloacetate. Our oxaloacetate will form citrate. So citrate basically will go through the series of reactions here. So citrate's going to lose a water molecule and gain another as it's converted into its isomer, isocitrate. Isomer we know is same formula, different arrangement of atoms. We see this enzyme. It's actually the only enzyme listed in this flowchart right here. Isocitrate dehydrogenase is the rate-limiting enzyme of the Krebs cycle. It determines how fast this system works, or how quickly this cycle goes through its 8 steps or 7 steps. All right, so after isocitrate, isocitrate is going to be oxidized, producing a 5-carbon alpha-ketoglutarate. During this process, it loses a carbon group, which turns into carbon dioxide, and 2 electrons are lost here, which reduces our NAD to NADH. So we've made 1 NADH here. Our next step, our step 4 right here, we're taking alpha-ketoglutarate and turning it into succinyl-CoA. During this process, we release another group of electrons, so reducing NAD to NADH. We also have another loss of a carbon, so we produce another CO2. And this action, or in this addition of a coenzyme. So we see a coenzyme A is added, so now we have succinyl-CoA. With our succinyl-CoA, it is lost, the coenzyme A is lost pretty quickly after it's added. So we're taking succinyl-CoA and turning it into succinate. We substitute that coenzyme A for a phosphate group. So our high energy bond is formed. So this is our phosphorylation part right here. It produces GTP. So GTP is guanosine triphosphate. That's GUANOSINE. So guanosine triphosphate. It's energetically equivalent to ATP, but its use is more restrictive in muscle protein synthesis, so used in muscle. So I'll show on the next slide how ATP and GTP work and produce energy, but this is our one site of ATP production. So we're now at our succinate. With succinate, we're going from succinate to fumarate. During this, 2 hydrogen atoms are transferred from FAD to make FADH. So FADH is a lower energy electron carrier. There's not enough energy in this step right here to reduce a NAD, but there's sufficient energy to reduce an FAD. I know that's kind of hard to understand. Just think of it as another hydrogen electron carrier. And so it's just another carrier. Again, it doesn't contain or operate with as much energy as the FADH does, but it will still make us more ATP in the electron transport chain. All right, so now, excuse me, now we go from fumarate to malate. All that happens here is a water molecule is added from fumarate to make malate. Now with malate, our last step in the cycle regenerates oxaloacetate. So oxaloacetate is losing, or to get from malate to oxaloacetate, we lose another group of hydrogens and electrons picked up by NADH. That is our process for one acetyl-CoA. So one acetyl-CoA equals 3 NADH, 1 FADH, and 1 ATP. But we have to double the products because we have 2 acetyl-CoAs from our 2 pyruvates. So basically, 1 glucose molecule equals 2 pyruvates, which equal 2 acetyl-CoAs, which equals 6 NADH, 2 FADH, and 2 ATP. I like this picture a lot, or this flow chart, this diagram. I don't know why. I think it looks clear, and it also shows all of the enzymes that are involved besides just the isocitrate dehydrogenase. So just kind of doing one more refresher of this. What we see at the top, pyruvate, what we have to know there is our production of 1 NADH and CO2. But again, CO2 is a waste product. Acetyl-CoA combines with oxaloacetate. We make our citrate here. I'm not going to ask you about any of these other enzymes. It's just kind of neat to see where they are and what they do. The only one I'm going to ask you about is isocitrate dehydrogenase, the rate-limiting enzyme that occurs between isocitrate and alpha-ketoglutarate. But you can also see the same things that are produced here. We see our NADH, and then another NADH between alpha-ketoglutarate and succinyl-CoA. You see the water molecule picks up that carbon. Again, we make that CO2. And then I do like down here how it shows the GTP and how this GTP basically donates its phosphate to ADP to make our ATP. So this is also what you'll have to know about the Krebs cycle is where the GTP is produced. First, what it stands for, guanosine triphosphate. It occurs between succinyl-CoA and succinate. I won't ask you about the enzyme though, so you don't have to worry about that. You just have to know where it's produced and basically what it is. So our only site of ATP production, then we go from succinate to fumarate. Again, there's our FADH. So with our FAD turning into FADH, it's just picking up 2 hydrogens. Like I said, this hydrogen carrier there's not enough energy present at this step to reduce an NAD, but there is enough energy present to reduce an FAD. So that's all you have to know. I'll explain in the next video how FAD and NAD are different and how they kind of produce different amounts of ATP once they get into the electron transport chain. So we'll get to that. And then again, we add a water molecule, we have malate, and then we lose some more electrons picked up by an NADH, we're back to oxaloacetate. So whichever flowchart you want to use to study this, you don't even have to know the order in which these molecules are produced. You mainly just have to know the products of production, which I'll talk about on the next slide, as well as rate-limiting enzyme and where that ATP or that GTP is produced. So what we want to do now is count up everything we have. So glycolysis, we produce 2 NADHs and 2 to 3 ATP, depending on what we started with. Our intermediate step, we produce 2 NADH. Krebs cycle, we have 6 NADH, 2 FADH, and 2 ATP. So thus far, I want you to think of, we just kind of started aerobic metabolism. So we're in that 2 to 3 minute mark of exercise at a pretty low intensity from 1 glucose molecule. This is what the body has produced. So now we have to take the NADH and the FADH and we have to make more energy because this is not sufficient enough. We need more. And so in part 2, the electron transport chain explains how we're making more ATP from these two hydrogen electron carriers. And so that will be explained. And that gets us to part 2, but we have to finish this with a video because videos help. I think they're important. So let me see first if this is the right video, and then we can get it up and going. Okay, so we'll finish this lecture with this video right here.
Turn that bite of food into a chemical that a cell can recognize and use as energy. The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a four-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of two distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, two ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the two 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistance. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain.
All right, so that video continues in the next part, so in part 2. So now you can watch part 2 whenever we go through the electron transport chain and discuss everything that happens there. So thanks for watching, and I will see you later.